In this series of videos, we will be looking at the structure of the male reproductive system. The presenter declares no conflict. The session objectives are listed in this slide and are also listed in the didactic notes. Male reproductive system functions in the production of semen and gametes, which is actually part of the seminal fluid and spermatozoa. It also produces sex hormones. Furthermore, it is involved in facilitating fertilization by delivering the gametes into the female reproductive tract. Some of the definitions that we will be associating with the male reproductive system are the testes, a system of genital ducts, accessory glands, as well as the penis. The accessory glands consist of the prostate, seminal vesicles, and the bulbourethral glands. The overview organization of the male reproductive system includes the testes, which are paired structures located in the scrotum. They have an important anatomic relationship to the covering and components of the scrotum. In histology, when you're in the lab, you will see one of those components which is covering the layer. It's known as the visceral layer or the tunica vaginalis. And you can see that here in this darker structure. Tunica vaginalis is a double layer of squamous epithelium which is shaped into a flattened balloon and locates right next to the testes. It allows the testes to be moved within the scrotum easily. And the testes themselves contain the seminiferous tubules, which is where the sperms are produced. Some of the other structures that we will be looking at includes testes, epididymis, prostate, vas deferens, the ejectory ducts, and the urethra. And all these are listed here, and you should review the anatomical locations in relation to each other. The accessory glands, which are listed here, are shown in red in this picture, are the prostates, seminal vesicle, the ejectory duct, as well as the bulbourethral glands. We will look at each one of these later on, so you don't have to look into it in great details here. This is a very informative slide, and you should spend some time studying this. The picture here in the left bottom here shows you what we have in the testes, as well as the duct system is involved with that, as well as its location within the overall anatomical structures of the male reproductive system. Now let's spend some time on this particular table, because it allows you to understand how the sperms are produced and transported from the testes to the penile urethra. The structures listed here, A, B, C, D, are what they refer to as intratesticular. So these are present within the testes, whereas E, F, G, H are located outside the testes, also referred to as the extratesticular. So this table shows you a structure like seminiferous tubule, its location in the testes, and what does it leads to, in this case, tubuli recti. So next, the tubuli recti, which is also located in the testes, but are the ends of the seminiferous tubules, then lead to Reed's testes. So without reading it all, you can start to appreciate that we are following a line going from seminiferous tubule to tubular recti, Reed's testes, efferent ducts, epididymis, vas deferens, ejectory duct, and then prostatic urethra. So this table should be a little helpful in understanding the progression as the sperms are traveling. So let us go through now each one of these structures in a little bit more detail. Testes are covered by thick connective tissue, which we refer to as tunica albuginea. And this itself then projects into the testes and form septa, which you have seen in multiple other organ systems, and divides this into little lobules. There's a posterior part of the capsule which thickens up and is known as the mediastinum, which is highlighted here in this red bracket. And this is the site where blood and lymphatic vessels are located within the testes. And also this is where you will find reed testes. Each of these lobules, which are shown here, are consistent of 1 to 4 seminiferous tubules. These are surrounded by connective tissue, and these are highly convoluted and looped tubules, as shown here, but in actuality, they are much more convoluted. The tubules end near here and near the mediastinum, and they end up in tubular recti, and then these tubular recti continuous with the reed's testes, and then which become the channels out from the mediastinum. We'll be looking at these structures in detail, so don't worry about looking at each one of them in great detail at this stage. This is a histological view of the testes. As you can see, it's not as clear cut as the picture that we were seeing earlier. And one of the reasons is that the plane of section. So in this case, for example, you are seeing tunica albuginea, you are seeing overview of the seminiferous tubules, and then ducts. In this case, these are the ducts of the epididymis, but nonetheless, these are not very well defined at this stage, but to understand that these structures are located next to each other. So looking at the seminiferous tubule in more detail, each one of these seminiferous tubule is about 30 to 70 centimeter in length, and as we said before, they are highly convoluted. The unique property of these tubules is that they are lined by the seminiferous epithelium. This is a very specialized epithelium composed of 4 to 8 cells in thickness, and which itself is then surrounded by these peritubular and interstitial tissue. The seminiferous tubules consist of 2 main cell types called Sertoli cells and spermatogenic cells. We will look at these in detail now. The Sertoli cells are pale in color, should be easy to identify in histological sections, but the other thing that gives it away are these oval nucleus, which is much bigger than any other nuclei that you will see within the section. Furthermore, they do have this very prominent nucleolus, which may or may not be visible in each cell in the section. The other property of the Sertoli cells is that they extend the entire thickness of the seminiferous epithelium. So this is the seminiferous epithelium, and then Sertoli cells are spanning all the way from the basal to the apical aspect of that epithelium. They do surround the adjacent spermatogenic cells, which are shown here in blue, and they are forming compartments for them within the thickness of the tubule. So this is just a quick overview so you can start to differentiate morphologically how the Sertoli cells appear compared to spermatogenic cells in the seminiferous tubule. The Sertoli cells themselves have many functions. One of the main functions they have is that they join with each other by gap junctions, and then by doing so, they tightly separate these basal cells, which are spermatogonia, from the developing spermatogenic cells, which are higher up here, by forming a barrier shown in red line here. And that barrier is very important to provide basically immunity from of these cells, the developing sperms, from the immune system itself. They provide support and nurture the developing spermatogenic cells, which are in this area here. They also excrete fluid into the lumen, which will be in this area here, that facilitates the transport of the fully developed sperm once it's released into the lumen here. In addition, they do produce some hormones like inhibin, for example, as well as androgen binding proteins, as well as anti-Mullerian hormone. These will be covered in more details in the physiology lectures. Furthermore, they act like macrophages of the testes by phagocytosing any leftover cytoplasm while the cells in the epithelium are maturing to become spermatids. So how do we look at the spermatogenic cells as they differentiate from the primitive cells into fully differentiated mature sperms? Well, as we've seen the seminiferous epithelium, they are very well sort of like ill-defined layers which are developing between the Sertoli cells. So in this picture here, for example, you're seeing the seminiferous tubule with a variety of different stages at which these cells are developing. Most of the time, you're going to notice the most immature cells under here, for example, referred to as spermatogonia. They are resting on the basal lamina of this tubule, for example. Whereas the most mature cells, in this case, spermatid, which are sticking out here, for example, projecting their tails into the lumen, are on the most apical aspect of the epithelium. In terms of spermatogenesis, we can look at the stages or the phases through which the sperm production goes. First stage is the spermatogonia, for example. Again, we are highlighting this at the bottom here, and these are the primitive cells from which all the cells then divide and differentiate to become mature sperms. They subsequently lead to primary spermatocytes, secondary spermatocyte, spermatids, and spermatozoa. Spermatozoas are the ones which are released into the lumen, and they are no longer associated with the Sertoli cells here. This slide kind of summarizes those phases that we have discussed. We would not be looking at histologically through that, so you don't need to spend too much time, but you should be able to understand this for other part of your block, for example. This is how the spermatogonia goes through the division to produce primary and secondary spermatocytes, and how the original mitotic division then changes to meiosis, which eventually gives rise to the haploid spermatids, and those then basically develop into the sperm. It is worth reviewing this part here, for example, because this is some of the pictures that you'll be looking at in the lab. The EM pictures do involve some of these structures. For example, once the cell has reached the spermatid phase, it is no longer dividing, but it is changing its shape extensively by remodeling itself. One of the things that happens is the formation of the acrosomal structures, which is the tip of the sperm. That's where it houses the hydrolytic enzymes for fertilization. Then the acrosome expands over the nucleus, and then nucleus itself condenses to the head region of the spermatid, and then the flagella structure starts to form. Mitochondria also starts to aggregate around the flagella, and then the cytoplasm within that started to diminish as the tail elongates. So these stages of sperm development is important, and you should understand that. But as far as histology is concerned, we would not be looking at it in great detail, except in the EMs in the lab. The other area where you should really be paying attention to is outside the seminiferous tubule, which the cells, so if this is the seminiferous tubules in the cross section, the cells which are laying outside, these cells are referred to as Leydigs, and they are found in clusters, and as I said, they're found between the tubules. They're easily identified as large, polygonal in shape, and they typically contain lipid droplets, which may or may not be visible in histological section. But one of their main function is to secrete testosterone, and this happens throughout the life cycle, and as well as the testosterone being important during puberty, it's also important in multiple aspects throughout, for example, for spermatogenesis, as well as in the accessory glands and duct development of the secondary sexual characteristics. So these cells are important when they're producing and secreting testosterone.